Cell membrane communicates with other cells, separate the cytoplasm cell parts from the outside environment, regulates the exchange of materials. Components and location of the cell membrane: phospholipids, cholesterol, integral proteins, peripheral proteins, and carbohydrate. Phospholipids, the main fabric of the membrane. Cholesterol, tucked between the hydrophobic tails of the membrane. Phospholipids. Integral proteins, embedded in the phospholipids bilayer, may or may not extend through both layers. Peripheral protein on the inner or outer surface of the phospholipids bilayer, but not embedded in its hydrophobic core. Carbohydrates attached to proteins or lipids on the extracircular side of the membrane. Fluid mosaic model, presented by Jonathan Singer and Grant Nicholson. In 1972, they presented the structure of cell membrane. The fluid mosaic model postulates that the membrane is composed of phospholipids bilayer with mosaic of various kinds of protein, cholesterol, and carbohydrates chain. Components of cell membrane: phospholipids bilayer, it has phosphate heads and two tails. Proteins. Carbohydrates attached in the cell membrane. Passive transport. Passive transport is a type of membrane transport that does not require energy to move substance across the cell membrane. Simple diffusion. It is the random movement of molecules of substance from a region of higher concentration to a region of lower concentration. This is the factor that affects diffusion. Extent of the concentration gradient. 2. Mass of molecules diffusing. 3. Temperature. 4. Solvent density. 5. Solubility. 6. Distance traveled. We can also apply in life process that involve diffusion. Plants absorb CO2 from the environment and it passes through the leaf. The lungs absorb oxygen through diffusion. Plants release the oxygen produced during the photosynthesis. Passage of amino acid of glucose into the blood. Facilitated diffusion. Facilitated diffusion is the process of spontaneous passive transport of molecules of ions across a biological membrane via specific transmembrane integral proteins. Osmosis. Osmosis is the spontaneous net movement or diffusion of solvent molecules through a selective permeable membrane, from a region of high water potential to a region of low water potential, in the direction that tends to equalize the solute concentration on two sides. Tonicity. Describe how an extracircular solution can change the volume of the cell affecting osmosis. Osmolarity. Describe the total solute concentration of the solution. Movement of water in a cell when placed in the solution of varied concentration. 1. Hypotonic solution. A hypotonic solution is any external solution that has low solute concentration and high water concentration compared to body fluid. 2. Hypertonic solution. A hypertonic solution is any external solution that has high solute concentration and low water concentration compared to body fluid. 3. Isotonic solution. Isotonic solution that contain the same concentration of water and solutes as the cell cytoplasm. Active transport. This involves the expenditures of energy by the cell. since the movement of materials goes against the concentration gradient, that is low concentration area to high concentration area. Protein carriers in the cell membrane are provided with energy by the cell to perform such function. Bulk transport. This process by which large substance or bulk amount of smaller substance enter the cell without crossing the membrane. Endocytosis. It is a general term describing a process by which cells absorb external material by engulfing it with the cell membrane. Three types of endocytosis. Phagocytosis, known as cell-eating, the type of endocytosis through which a cell takes in a food particles. Pinocytosis, known as cell drinking, the type of endocytosis by which a cell absorbs small particles outside and bring them inside. Receptor-mediated endocytosis, process by which cells absorb metabolites, hormones, proteins by the inward budding of the plasma membrane. It is a form of endocytosis in which receptor protein and the cell surface used to capture a specific target molecules. Exocytosis. Exocytosis is the fusion of secretory vesicle with the plasma membrane and resulting in the discharge of vesicle content into the extracircular space and the incorporation of new protein and lipids into the plasma membrane.